Okay, did you think about why magnification high and color absent in a retouch? Why do we need to have color in why are we staining things, essentially, is the question. Why? Why do we stain things? Far smaller than those of visible light and provide resolution. Electrons have no property analogous to color, and electron micrographs are basically density maps rendered in black and white. So that's a little fun part about electron microscopy, which we don't have, but staining. Okay, staining. Gotta stain it because otherwise we can't see it. So if you didn't stain your specimen, it would look like this. And let me tell you, friends, you are not going to be able to see that. I barely have the skills on a good day. I'm going to go blind trying to focus this. It's going to take forever. I'm going to be frustrated. And when I look up, I will see stars for days. Nobody wants that. It's too hard. Absolutely not. We're not doing that. So what we're going to do is we're going to stain our bacteria. Not everything needs to be a gram stain, but we need a basic stain just so we can see that thing in the microscope. Do you see why this is going to be impossible to focus? Forever. It will take you forever. Nobody wants to do that. So you got to prepare that thing from staining. Now, you are going to spread this culture over the slide. You are going to put it on your slide. And what are you not going to do? You're not going to blow on it. Because we're not blowing on stuff in microbiology. We are not blowing on things. You're going to want to blow on it because it's going to take a while to air dry, but you are not going to blow on it. You're not going to do that. You're going to air dry it, and then you're going to pass it through a flame. You're going to pass it. It's glass. You're not going to hold it over the flame until it blows up. That's not what we're doing. You're just passing it over. You're just dry and fixing it, right? It's a pass. Okay, and now you're ready to stain it. Here's some stains. And my friends, we're going to do all of these stains in class because that's what lab is for. We are going to do some simple stains, which is what we're going to do the first week. Then we're going to do some differential stains. We're going to gram stain it. We're going to use some India ink. We're going to use some methylene blue. We're going to use acid fast stain. We're going to look at your flagella stain, and nobody's in this class is going to look like this. It's just not. Sorry. I don't know why this one looks so awesome, but I've never seen one that looks that good. So dial down your expectations. Okay. Principles of staining. Essentially, acidic dyes stain alkaline structures and basic dyes stain acidic structures. Acids and bases. So we will very specifically do an acid-base stain. So the beauty of this really is like you can be in the middle of the jungle in a developing country with a microscope and a generator and some stain, and you can figure out what potentially a disease is. Okay? I promise you. I know that when you're in the middle of the unknown project, you're going to feel like that's not possible. But I assure you, this is why microbiology is so important, because you can do some of this stuff without, having to run a lot of expensive tests. So there's an acid base stain that we will do. We will do some simple stains. This will be on Tuesday. We're going to start here with the simple stain, and it's going to look like this, right? It's just you're going to be able to see it because it's blue, and you're going to be able to focus it. Now, also, we are going to do a gram stain, and we are going to do some of these called differential stains. Essentially, you have a gram negative and a gram positive. You're going to put some crystal violet on that thing. Okay, right? Then you're going to add iodine. Okay, it's going to turn purple. Fair. Then you're going to decolorize it. You're going to bleach it out. Well, the one that has all of this peptidoglycan is going to absorb that iodine and crystal violet. And it's like, no, you can't have it back. No, I'm not bleaching. No, it's mine. It's going to absorb in there. And so then after you decolorize it, you're going to put a counter stain of pink. Well, this isn't going to turn pink because it's already purple. These are going to turn pink. Okay? That is essentially what we're doing. And it looks different if you're doing an acid fast or an endospore, but that's essentially what you're doing. You're going to put a thing on it. You're going to put another thing on it to make it a color. Then you're going to decolorize it, and you're going to color it again so that you get two distinct colors. Right? This is honestly harder for people that don't dye their hair. If you dye your hair, you kind of know how this thing works. They are going to look kind of like this. Okay? Yeah, it's going to look... All cells are stained purple, so if you were to look at it in between these processes, which you're not going to do because that's going to make a mess, and it's going to turn the microscope purple. No one's doing that. So then you're going to flood it with iodine. Again, everything looks purple. Then you're going to rinse it with ethanol and acetate. Okay, great. Now these, you can kind of tell, are going to be the gram-positive. But where are the gram-negative? I can't even see them. So now I have to stain the ones that are clear. Do you see why that last step with the safranin has to happen? So now I can see those guys because they're pink. Before, all I could see was the gram-positive because they still had that purple. But now I want to restain the clear ones so they're pink. So now I can definitely see these are pink, these are purple. Do we understand what's going on there? Okay. Here's an acid-fast stain. Yours is, honestly, these are fairly easy. Yours is going to look maybe like this. Your endospore stain is probably not going to look this good, but it'll look kind of like that. So it's fun. I like a good stain. It's pretty fun. Two kinds of stains. There's eosin, there's silver stain. You can do these for very specific, once you narrow down your bacteria, you're like, oh, I need to do a flagellar stain. I need to do a different stain to see, is it acid fast? Does it have an endosperm? Right? These are things you're going to need to know. Here is a really great negative stain. I've never seen a negative stain look this good, but surprise me. Simple stains, you're going to use crystal violet. All of these stains are in the lab. They're either in the refrigerator or on the bench. And these are all the ones you're going to do as you start learning how to differentiate the bacteria. And again, you get to do these with just a microscope, which is pretty fascinating. They still stain stuff for electron microscopy, but we are obviously not doing that because we don't have an electron microscope. And here's a little staining video.
Utilize stains to increase the contrast and visibility of the specimen, as well as to learn more about the features of the specimen. There are three main categories of stains used by microbiologists: simple stains, differential stains, and special stains. Simple stains are made of a single basic dye that interacts with the biological sample and stains it. After treatment with the dye, the sample is rinsed with water, leaving the stained sample. Simple dyes are used to determine size, shape, and arrangement of cells. Crystal violet is a purple.
Those are all the same kind of stains, so I'm going to just fast forward. Now we're going to
Used in microbiology are differential stains. Differential stains use more than one type of dye and can be used to distinguish between different structures. The gram stain is the most frequently used differential stain. It's used to distinguish between gram-positive cells, which have a thick outer cell wall composed of peptidoglycan, and gram-negative cells, which have a thin outer cell wall. The procedure of gram staining involves 7 steps, during which the samples are stained with crystal violet dye, treated with a mordant, rinsed, treated with a decolorizing agent, and counterstained with safranin.
Did you see that right there? Our gram-negative is now unstained. But you're not going to be able to see that thing, so we're putting some pink in there. Now you're going to be able to see it under the microscope.
Gram-positive cells are stained a deep purple color from the crystal violet dye, but gram-negative cells are decolorized and then stained pink by the safranin. The acid-fast stain is a useful differential stain because it only interacts strongly with a subset of bacteria in the genus Mycobacterium that have a waxy material in their cell walls, as well as the pathogenic strains of the genus Nocardia. The acid-fast staining method involves several steps, during which the samples are stained with carbofusion while being heated over steam, cooled, decolorized with hydrochloric acid, and counterstained with a simple dye such as methylene blue.
You get to use hydrochloric acid. What's not to like? There we go.
Some bacteria produce endospores. Endospores form inside the cytoplasm of the bacteria and are highly resistant to desiccation, heat, and radiation. Their cell walls are impenetrable to most chemicals. The endospore stain uses heat to drive the dye malachite green into the endospores. After heating with malachite green, the slide is rinsed with water to remove